fiber types first. The more intense the exercise gets, the more type 2 muscle fiber types are recruited for the activity. And then, so if an individual has more type 1 muscle fiber types, the depletion of muscle glycogen might be different. Figure 5.9 shows on the left, it shows fast twitch muscle fiber types in white, and it also shows slow twitch muscle fiber types in dark. And if you look over to the slide on the right, you'll notice that the fast twitch muscle fiber type is still stained red. That means that that fast twitch muscle fiber type, well into that bout of activity, still holds on to its muscle glycogen. You'll notice that all of the slow twitch muscle fiber types that surround that fast twitch muscle fiber type are void of muscle glycogen. So an individual that has predominantly slow twitch muscle fiber types will burn through their muscle glycogen faster than an individual that has predominantly fast twitch muscle fiber types. So on slide 43, the utilization of muscle glycogen for prolonged exercise is not enough, so your body and your liver is going to have to produce glucose through glycogenolysis or gluconeogenesis. As the muscle glycogen concentration decreases, the amount of work that the liver is going to have to do to replace that need for glucose in the central nervous system and the muscle is going to have to increase. And it could be the case that muscle glycogen depletion leads to hypo, too little glucose in the emia, which is the blood, which is going to potentially lead to fatigue. So it could be the case that depletion of muscle glycogen and blood glucose is also an indicator of fatigue. If you look at figure 5.10 on slide 44, it shows different muscles. It shows in red level walking, in blue uphill walking, and in yellow is downhill walking. And in each instance, the gastrocnemius is requiring more, is utilizing more glycogen than the other muscles, which is an indicator that the gastrocnemius is utilizing more glycogen than the other muscles during that bout of activity. So when we're discussing potential causes of fatigue, that gastrocnemius muscle may fatigue faster because of a reduction in muscle glycogen than the other two muscles. So it could be one of the muscle groups that are specialized in a movement could also affect the amount of fatigue. It could be the case that because figure 5.10 is a running exercise, remember the gastrocnemius fires isometrically almost constantly in order to keep the Achilles tendon taut. So during a run, you can hit the forefoot and use the Achilles tendon as a spring. So that's why the contribution of the gastrocnemius during run is so high. And also be aware that the vastus lateralis, which is a quadricep muscle, is also firing eccentrically to absorb the ground reaction force during running and also create more of a leg swing during that gait. So that's why there's an increase in activity for both of those. Okay, so let's go to slide 46. There are potential causes of fatigue due to metabolic byproducts. We talked about how the breakdown of adenosine triphosphate is going to result in a large amount of inorganic phosphate in the blood, the concentration of blood and plasma. So that increase in phosphate is a rate limiting factor to the creation of ADP, ATP from ADP, and that potentially could lead to fatigue. In regards to heat, as body temperature increases and core temperature increases, that could also lead to fatigue. Lactic acid production as a product of anaerobic glycolysis could also potentially lead to fatigue, as well as the increase in hydrogen atoms that are caused by a breakdown in lactic acid. The breakdown of lactic acid is going to create lactate and this a hydrogen ion, and that positive hydrogen ion is going to create an acidic nature in the bloodstream, which is going to potentially negatively impact the rate at which calcium is released from the sarcoplasmic reticulum, which could have a negative impact on contraction, which could also be one of the thought of causes for fatigue. In regards to heat, heat changes the metabolic rate. It increases the rate of carbohydrate utilization and makes it so that glycogen depletion occurs faster. So muscle temperature is correlated with a faster oxidation of glucose and might make it so that the muscle function reduces. Individuals who exercise at 11 degrees Celsius, which is a little bit low that I thought 51 degrees Fahrenheit, have the longest time to exhaustion. So muscle temperature and body temperature may have something to do with fatigue. On slide 48, it shows individuals' time to exhaustion at various temperatures: 4 degrees Celsius, 11 degrees Celsius, 21 degrees Celsius, and also varying humidities. So at 24% humidity and 51%, 51 degrees Fahrenheit seems to be the ideal conditions for an individual to avoid fatigue. So hot summer days are not necessarily the best to run a marathon. Look for a cool fall day. Slide 49, lactic acid accumulates during high intensity exercise, especially short term high intensity exercise. And if you can't get rid of the lactic acid, then what happens is much of it is going to be converted to lactate plus this hydrogen atom. And the hydrogen atom is going to increase the acidity of the muscle. And because of that, there's a reduction in the ability for calcium to be released from the sarcoplasmic reticulum, and it also makes a hyperacidic situation in the blood, and that can lead to cell death and cellular membrane dysfunctioning. A pH less than 6.9 makes it so that the body cannot break down carbohydrate and glycolysis. And that if that's the case, ATP is not going to be able to be generated. So low acidic natures of the blood stop blood glucose from being broken down. As you exercise, this is figure 5.12 on slide 50. When you sprint, there's an increase in the production of lactic acid, which is going to decrease the muscle pH, and that muscle pH is going to increase back to normal levels, but over a period of time, like 20, 25, 30 minutes. So that could be the cause of fatigue that occurs longer than the bout of exercise. Slide 51 talks about the potential neural transmission interruption that could cause reduction in fatigue. First of all, the central nervous system functions and gets fuel strictly from glucose. So if your body doesn't have glucose, then the central nervous system can't function, and it's going to have a negative effect on the distribution of action potentials. It could also be the case that there's a reduction in the creation of acetylcholine, and the way that acetylcholine goes across the synaptic cleft, and it could potentially affect the resting membrane potentials of the post and presynaptic cleft neurons, and might potentially result in a reduction or an inhibiting the release of calcium from the sarcoplasmic reticulum. But the central nervous system is not necessarily fully understood in its roles with fatigue. One of the things we've all experienced, that we're fatigued, we're doing that Spartan race, that Tough Mudder, we're 26.3 miles into it. Not we, I didn't do that, but 26.0 miles, and you got to go that extra 0.2 of a mile, and somebody comes up to you and they motivate you and they motivate you and you motivate you, and it seems as though that motivation has a lot to do with a reduction of central nervous system fatigue at that moment. So the impact of psychology and motivation and things like that are not 100% understood yet. So it could be that the central nervous system does play a role in fatigue, but the exact mechanisms by which it does so and by which it is affected or infiltrated still needs research. So you guys all have potential doctoral theses. Um, uh, we're on slide 53, talks about muscle soreness. There's many different potential causes of muscle soreness. There could be acute causes of muscle soreness during and immediately after, and there could also be muscle soreness that occurs like 2 or 3 days later. That's referred to the DOMS, delayed onset muscle soreness. We'll talk a little bit about each of these and potential reasons why. Acute muscle soreness could be due to the increase in metabolic byproducts such as the hydrogen ions and the change of the blood concentration to a more acidic nature. It could also be due to micro damage in the muscle during the exercise, which is leading to an attraction of neutrophils and white blood cells to the area, which is causing micro swelling and acute swelling. It should be the case that this acute muscle soreness is able to disappear within a few hours, especially if you have an active cool down. What happens during an active cool down, a 5-minute cool down after your bout of exercise, is you continue to utilize the muscles, but the muscles then aren't creating as much lactic acid. And if that's the case, then blood that doesn't have lactic acid can flow into the muscles and return the pH of the muscles back to a normal or more resting level. So the results of that micro soreness should potentially dissipate. Delayed onset muscle soreness can potentially occur 1 to 2 days after exercise. The major causes are not 100% known, but it could be the case that eccentric muscle contractions cause micro damages, and those micro damages are attracting neutrophils and the breakdown of tissue through phagocytosis and all sorts of stuff. And it could be that the micro swelling and the breakdown of the tissues is leading to potential soreness. The structural damage to the muscle is indicated by an increase in enzymes. The concentrations of some of those enzymes increase to 2 to 10 times amounts during heavy training. And as you graph delayed onset muscle soreness, it seems to be correlated with those increases in enzymes in the blood. There's also a severe disruption of sarcomere integrity during high intensity exercises. I don't know if there's a slide for that. It doesn't seem as though there. Let me see. Let me go quickly. No, there's no slide that shows that, but there's a slide in the book that shows. Sorry. There's a slide in the book that shows a picture of Z discs and actinomycin on a little slide before an activity, and then the individual runs a marathon or something. I can't remember what the activity was, and it shows another slide, and all of the Z discs and all of the thin and the thick filaments are seeming like lysed and split in half and broken up, and there's a large amount of damage to the muscle. But muscle damage makes it so that the neutrophils and the testosterone is released, and oftentimes it's going to result in hypertrophy and muscle strength and muscle growth. So the muscle damage, the micro damage because of exercise and the delayed onset muscle soreness that is symptomatic of that muscle breakdown and that muscle damage is the precursor to hypertrophy. So I mean, it seems as though managing the delayed onset muscle soreness is necessary, but it actually seems to be a precursor to muscle growth and strength and development. So it could be the case that inflammation, we're on slide 57, that inflammation increases soreness as you do exercise and cause stresses to the muscles, and they go through micro damage. White blood cells are brought to those sites. Increase white blood cell count is associated with an increase in soreness, but it could be because of inflammation. Damaged muscle cells attract neutrophils, and the neutrophils are white blood cells that break down certain damaged cells, and they release other cells that are going to help build those cells back up. And it could be the case that these neutrophils and the breakdown of those muscle cells result in a stimulation of nerve cells, and that could potentially lead to a perception of pain. Um, the DOMS is potentially occurring because of a high tension in the muscle, especially during eccentric muscle contractions. That high tension in the muscle is kind of like pulling the muscle apart, is fraying the muscle and causing structural damage to the muscle and the cell membrane. And then the cell membrane is going to have to release some type of signals that's going to allow for a hormone release that's going to result in some type of protein synthesis at some later point so that those tissues can heal. After a few hours of that membrane damage and the disruption of the calcium in the sarcoplasmic reticulum, there is going to be an increase in the circulation of white blood cells and neutrophils so that the the tissue can get healed. Then there's going to be products of the breakdown of those particular those cells like histamines and kinines, and that's going to stimulate pain in the nerve endings around the muscle that's going through this micro damage and healing process. And then there's going to be a rush of fluid and nutrients and electrolytes into the micro swelling or the micro damaged muscle, and that's going to create swelling, which could also lead to an increase in the perception of soreness. We're on slide 60. Delayed onset muscle soreness is associated with a reduction in muscle force generation from anywhere to 2 days to a couple of weeks afterwards. So there's going to be a loss of strength because of delayed onset muscle soreness. It could be because of that physical disruption of the muscle. It could be because the actin and myosin interaction is not occurring, or because the neural transmission of the action potential is fatigued and not occurring effectively, or it could also be because the actin and myosin thick and thin filaments and they've actually been damaged, so you've lost a little bit of actin and myosin protein, so the muscle contractions can't be effective. They need to go through protein synthesis, and testosterone and growth factor needs to be sent to those muscle fibers, and hypertrophy needs to occur. After hypertrophy occurs, the muscle is going to be stronger than it was before. Figure 5.15 shows that days after your injury, what parts of that particular the mechanisms for soreness are still at play. When you've done intense exercise, it shows here that up to 3 days after that exercise, you could still have physical disruption of the actin and myosin and the sarcomere. Up to about 1, 2, 3, 4 days, you're going to have the excitation coupling failure, so the disruption of the calcium sarcoplasmic reticulum and the actin and myosin intertwining will still be potentially interrupted up to 5 days, and there's a reduction after 3 days, but you'll still see that the blue column is still relatively high. 5 days after, and about 2 weeks later, that will have subsided. And you could also have the contractile protein loss, the actin and myosin, that could also remain a factor up to 3-4 weeks after that bout of activity. So. In order to have effective training, you have to be able to reduce the symptoms of delayed onset muscle soreness. So, want to make sure that we're doing things like progression and starting out with low intensity. So the guys in the gym that I had start a strength and conditioning program, we did about a 5-week introductory period where we taught them all the exercises. We started with bungees or unweighted bars or even PVC pipes instead of dumbbells and things, so that we could treat them proper technique. We could start with very low weights so that we wouldn't shock their muscles. Although there are some suggestions that what you do is the very first day or week of training, you do very high intensity to push those muscles through delayed onset muscle soreness, so that the athlete experiences almost like the worst pain that they're going to experience at first, and then subsequent bouts of training seem to be less and less invasive. But through progression and starting out with low weights and slowly increasing the intensity of the exercise, it seems as though that's one way to minimize delayed onset muscle soreness. Muscle cramping is another particular limiting factor in exercise, especially in high heat, and it could also lead to the completion or the early stopping of the bout of exercise. It could be the case that it has to do with neuromuscular control. It could be the case that it has to do with the excitation of the Golgi tendon organ that is sending an excitation, an inhibit, a response to the spinal cord that sends an excitation response to the muscle. And it seems as though that muscle cramps or exercise-associated muscle cramps can be relieved by stretching. I believe strongly in foam rolling and also hydration. Heat cramps are associated with a large amount of sweat and electrolyte loss, so make sure that if you are exercising in the heat, not only are you concerned with replacing fluids, but you're also concerned with replacing electrolyte and any other electrolytes that you're expending during sweating and things like that. Make sure that you keep the body at normal temperatures or reasonably normal temperatures. And if you are experiencing heat cramps, make sure that you have a high sodium solution. Like put sodium chloride in water and glucose solution, and do ice and particularly a massage will help get rid of the symptoms for most individuals. Slide 65 is a video that talks a little bit more about what we just learned, and I'm going to call it a wrap.